Aleksandra Pękowska graduated from the University of Lodz in biology in 2006. Next, she joined Dr. Pierre Ferrier's lab in Marseille, where she described chromatin signatures of active enhancers and tissue-specific genes. In 2011, Dr. Pękowska joined the group of Dr. Wolfgang Huber at EMBL in Heidelberg. She found that the loss of pluripotency coincides with a gain of chromatin loops. In 2016, Aleksandra Pękowska joined the team of Dr. Rafael Casellas in National Institute of Health in Bethesda, and she described a unique chromatin structure linked to the control of gene expression. In 2019, Dr. Pękowska opened the Dioscuri Center of Chromatin Biology and Epigenomics at Nencki Institute of Experimental Biology in Warsaw. She combines omic tools, CRISPR-Cas9 and stem cell models to understand the genetic basis of astrocyte functions and the implication of genome topology and transcriptional regulation. That's the shortest version of CV, and I am very happy and proud to host today Dr. Aleksandra Pękowska. Aleksandra, if you could switch on your camera and microphone, let's connect. Good afternoon.
Good afternoon.
And we can start. So please share the presentation and please start. Digital Zoom is yours.
So thank you very much for this kind introduction, and I hope everything is nicely visible. So thank you very much for the introduction and for giving me the opportunity to discuss the topic that has been in the center of my research ever since my postdoc. And we will be talking about the intertwined relationship between how genomes are organized in the three-dimensional space of the cell nucleus, and how this organization is related to gene control. So one of the central questions in biology is how a single genome operates to ensure the development and the establishment of cell identity during development and the generation of hundreds of cell types and tissues in the mature organism. And to understand this phenomenon, we need to consider what cell identity is. So data generated over the past decades clearly shows that every cell expresses a unique set of genes. For instance, a neuronal cell keeps neuronal genes active. These include genes that encode proteins related to synaptic function, and while keeping other genes silent. And for instance, a red blood cell will silence synaptic genes. However, it will activate genes that are important for its functions, namely hemoglobin. So we can schematize it in a very simple way. A nerve cell will silence hemoglobin and express neuronal gene, while a red blood cell will do the opposite. And the central question is how this is obtained molecularly. So we know that cell type specific gene expression is ensured by the activity of dedicated regulatory elements in the genome, the so-called cis regulatory elements. These include promoters that promote gene expression, enhancers that impact it by increasing it, insulators not depicted on this very short simplified schematic. And the enhancers are typically the most important factors for this regulatory functions of the genome. Enhancers are relatively short sequences, and what is critical for their activity is the fact that they harbor multiple binding sites for transcriptional regulators, so-called transcription factors. And it's by binding these transcription factors that enhancers can enhance particular gene expression. So over the past years, it has been also appreciated that there is particular chromatin arrangement, or not really arrangement, the particular chromatin modifications that are that witness or can tell us about enhancer activity. These include histone 3 on lysine 4 monomethylation, histone 3 on lysine 27 acetylation, and these marks are particularly enriched at these elements. In the past, we also discovered that particularly active enhancer elements regulating important genes for during development are also enriched in H3K4 trimethylation mark, which is typically found at highly active promoters. So enhancers are also quite atypical regulatory elements because they exert their function over vast genomic distances. They can be located up to megabases away from the genes that they regulate. And it's actually a big question in the field, how the specificity of enhancer promoter interaction is obtained. In other words, how do enhancers know, so to say, which genes they are supposed to regulate? And this in the context of complex chromatin architecture. So in order to understand this, we have to understand how chromatin architecture is established and how this quite long molecule, up to a couple of dozens of centimeters, can give this particularly beautiful, in my opinion, arrangement of chromatin in the cell nucleus, and how this arrangement is related to functionality and interactions between regulatory elements. So fluorescence in situ hybridization and immunofluorescence studies shed a light onto basic principles of chromatin organization in the interface. We know that chromosomes occupy dedicated territories, so they were deemed chromosome territories. Chromosomes can intermingle, but to a not very profound degree. And what is also quite fascinating from a functional standpoint is the fact that on this framework of chromosomal territories, we have activities that are also clustering. Transcription, for instance, occurs not really uniformly throughout the nucleus. And we know that there are regions enriched in polymerase II. They were deemed transcriptional factories. We'll talk a bit about this toward the end of the lecture. Also, DNA replication occurs in spatially confined regions in the chromosome, in the genome. So the major now question is, of course, what drives the formation of these structures and how this formation can be stabilized and regulated. So during my talk, we'll dwell deeper into some of the recent data that helps to better understand the following phenomena and address the following question. How is the interface genome structure obtained? And how does the genome structure participate in transcriptional regulation? So to understand this, of course, technological development provided us with tools that allowed us to understand better these questions. And the technologies that I will be talking about during this talk are primarily related to nuclear ligation assay, chromatin conformation capture, and HICE. So the nuclear ligation assay is derived from actually cyclization enhancement. And this was an original idea published back in the 80s that has been developed over the past years to give us this high throughput sequencing-based technologies. But it's really amazing how the concept that was coined quite long ago is still helping us to understand really cool questions in nuclear biology. So these technologies rely on the fact that, on the observations that active regulatory element dialogue is most probably related to establishment of physical proximities between sequences. So in essence, what we are going to hunt for are physically proximal regions in the genome. So it's very nicely depicted in this electron micrograph. You can see like here two sequences that are really very close to each other. So the original idea was to cut the genome once we fix this by, for instance, formaldehyde, to cut the genome by restriction enzymes, and then ligate the sequences that were close to each other using ligase. And already back in the 90s, it was shown or suggested that active regulatory elements that can be very far apart in the linear sequence of the genome can establish a physical proximity with each other. And then this establishment correlates with transcriptional onset. So years later, Jobdecker and colleagues established and developed the so-called Chromatin conformation assay. And almost a decade later, R.S. Lieberman-Naden, together with Jobdecker and colleagues, established the so-called high C. So high C, as mentioned already in this nuclear ligation assay, uses the same principles. So basically, in this assay, we cross-link proteins to DNA also, with formaldehyde. So this fixes and preserves spatial proximities between regions in the genome. Then we cut the genome by a restriction enzyme that generates cohesive ends. And we fill in these ends with biotinylated nucleotides. Then we perform what is called the proximity ligation step, which basically is just incubating the complexes with ligase. Then we remove proteins, isolate DNA, shear it by sonication, and pull down those sequences, those hybrid junctions between regions that are not really necessarily close in genomic sequence, but are close in space. This can be two different chromosomes. And we pull down these chimeric ligation products by streptavidin beads. Then we append paired end sequencing adapters and we sequence. And basically then we bin the genome or not, and count how many times a particular sequence was sequenced with the other sequences in the genome. So this gives us this maps of proximities. So here is the beginning of the chromosome. That's the end of chromosome 2. Here is a map for neuronal progenitor cells in mouse. And what you see is the whole chromosome interactome. And as expected, sequences that are close to each other in genomic scale, they also are very red here. So this means that they are very frequently sequenced in the same ligation fragments, which means that they were close in space. However, sequences that are far apart on the same chromosome, they display lower signal, but it's not zero. So basically, many sequences for chromosomes fold. They basically form structures where even very remote fragments within the same chromosome can be found close to each other. What is also striking in these maps and was observed by Lieberman-Naden and colleagues is the fact that chromosomes, this is not random how chromosomes fold. So we can observe here a sort of checkerboard pattern of interactions where some regions interact preferentially with other regions on the same chromosome and very rarely with some of the remainder regions. So this corresponds to partitioning of the genome into euchromatic regions and heterochromatic. There's also much more complexity to this. We will not really discuss this during this talk, but there's a very nice area of research investigating forces that drive this compartmental context. They were deemed compartments. Instead, during my talk, we will discuss more interactions that occur like here, close to the diagonal within genomic intervals of up to maybe 2-3 megabase in length. And we will do so because most of the cognate promoter enhancer pairs, they are separated by such distances. So when the Heidelberg maps were first sequenced to a great depth, couple of labs in parallel, Bingren, Edith Hurd, and Giacomo Cavalli labs discovered a partitioning of the genome into those topologically associating domains at this length scales. So what you can see here is gene models, and here this Heidelberg maps where again red corresponds to closer spatial proximity. So what you can see is that we have those domains of preferential contacts, and sequences within such domains contact each other more frequently than across the domains. So it was also shown by these papers that topologically associating domain boundaries are formed by CTCF factor. We'll talk much more about this. It's the 11th zinc finger transcriptional regulator and the only known mammalian insulator to date. So the fact that they were delineated by CTCF binding sites and the fact that many cognate promoter-enhancer pairs occur within a single TAD led to a proposal that TADs could act as genuine functional units of genome organization. So first we needed to show what they correspond to in single cells. So there was a beautiful development of fluorescence in situ hybridization coupled with super resolution microscopy by Zhuang lab. And in this technique, the authors used oligopaints to gradually paint the whole domain and monitor the spatial distances between the probes in a single cells and single alleles. So what they've shown is that topologically associating domains can be scored in single cells. They really act like structures. However, they are quite dynamic from allele to allele. But when you average them out across many hundreds or thousands of chromosomes, you see a similar profile than the one that we see in Heidelberg. So Heidelberg very nicely reflects the average profile across those stats. So there's also single cell Heidelberg. I will not discuss this during this talk, but this technology also in principle could allow us to monitor TADs and the structures in great details in single cells. So there is a substantial heterogeneity of these structures. However, on average, TADs correspond to what we see by Heidelberg. It didn't tell us, though, the mechanisms that drive the formation of TADs. And the improvement of Heidelberg and the development of the so-called in situ Heidelberg by R.S. Lieberman and colleagues allowed us to understand better how TADs are formed. So using these ultra-high depth resolution maps, R.S. Lieberman, sorry. I'm sorry, I apologize. R.S. Lieberman and colleagues discovered that frequently TAD boundaries form this very strong interaction, reminiscent of genuine chromatin loops. They uncovered the existence of thousands of such loops in the genome, around 10,000. And when looking details into CTCF binding and its profile at the anchors of these loops, the authors realized that, that CTCF binding occurs, so the two sites that form a loop in chromatin, they are frequently facing each other. So this was termed the convergence rule. And why did it help us to understand chromatin structure and how it's obtained? Well, if you consider the sizes of TADs, which are up to a couple of hundreds of nanometers, and the size of CTCF itself, which is a tiny molecule of around 5 nanometers in diameter, it becomes obvious that one cannot explain the formation of this structure by a simple 3D looping mechanism or even 3D diffusion, because then how would you obtain the convergence? How would you enforce the preferential interaction between two sites that face each other? So the only way of obtaining it would be a model that would use a 1D scanning mechanism, where there would be factors that bind to chromatin and basically slide along it and then stop on CTCF sites that face them. So what could be those factors? So back in the beginning or in first decade of 21st century, several labs noticed that CTCF co-occupy sites with cohesins. And cohesins are discovered by Kim Nasmith are heterotrimeric complexes that ensure sister chromatin cohesion in mitosis. They bind to DNA topologically, or pseudo-topologically, it's unclear, and are composed of structural maintenance of chromosome 3 and 1 molecules, along with RAD21. They are loaded onto chromatin by the NiPL-MAU2 heterodimer. And studies in Drosophila and later in humans revealed that cohesins are important for transcriptional regulation. It was unclear how, but removal or hampering of their correct loading onto chromatin by, for instance, NiPL mutation leads to multiple abnormalities, and, for instance, Cornelia de Lange syndrome. So in the past, we found that removal of the NiPL factor and hence removal of cohesins from chromatin results in abolishment of TAT formation. Others did this, we did this in the mouse. Sorry. Others did that in other settings and confirmed that very beautifully. So we now know that TADs are basically formed by cohesins and CTCF. And how do we now put all this data together and to understand TAD formation? And the current model posits, the loop extrusion model posits that cohesin loading onto chromatin by the NiPL-MAU2 complex leads to a formation of a complex that allows, I'm sorry, I hope you hear me. So the cohesin complex is loaded onto chromatin by NiPL-MAU complex, and then extrudes a loop. And then the movement of cohesin is stopped by binding, or when it encounters CTCF, bound to a motif that faces the arriving cohesin complex. Then the other side can still continue extruding the loop. However, this process stops when the other side encounters another CTCF site that is, again, facing the upcoming cohesin, which results in the formation of a loop between the two convergently oriented CTCF binding sites. Now, the development of really cool technologies on in vitro, though, still allow us to monitor and observe these motor protein extruding loops in living in life circumstances. So here is an example of another loop extruder that is loaded onto chromatin fiber that is attached to a DNA fiber that is attached to a microscopic slide. And just by monitoring fluorescence, you can see a growing loop that is produced by these big motor proteins. So, so in general, right now, the model posits that genomes are organized into those topologically associating domains that frequently harbor cognate promoter enhancer pairs. They are delineated by CTCF and produced by cohesions. So in our past work, we were struck by one of the features of high C maps that weren't really well described. We deemed them architectural stripes. And in these structures, we have one of the edges of the domain that is highly interactive and basically builds strong contacts with the entire domain. So we uncovered those architectural stripes in virtually all the high C maps that we saw in human B cells, mouse ES cells, or mouse B cells. And we were wondering what they are linked with. So first we needed to establish a method to score these structures from high C maps, which wasn't really as simple because these structures are somewhat weak as compared to other pixels. Our eye has the tendency to very nicely cluster them, but it was much harder to tell that to a computer, but we managed, of course. And so we have this stripe color. And then we were able to compare stripes across different conditions and relate them somehow to loop extrusion and perhaps transcriptional regulation. So we will define this stripe anchor as the anchor that produces those interactions. And this would be a stripe domain. We won't talk about this more. Let's focus on anchors. So when we compared stripe anchors in B cells and in ES cells, we found that these structures were relatively cell type specific. So there is many more anchors that we described in B cells, and we'll go back to this, than in ES cells. And then when we looked at overlap between critical features of chromatin activity, such as enhancers or super enhancers, which are simply clusters of enhancers, and active enhancers, and enhancers that are poised, so they will be active in other state, we found a strong overlap and very strong enrichment with the stripe anchors. So stripes are cell type specific and overlap active regulatory elements. So then we knew that these structures are somehow related to transcriptional regulation. Then we wanted to understand what drives their formation. So first we looked at the distribution of histone H3K27 acetylation, which, as you know already, is associated with active enhancers. And we found that the stripe formation follows the enrichment of this modification. So at the domains that we have stripes on both edges, there is very high enrichment of this modification across the board. At domains that form stripe only on one edge, the edge that forms the stripe is enriched more with this modification than the edge that does not form. And then we had a look at the location of cohesion loading factor, NIBL. And we found that NIBL was in fact following the enhancer location across the domain. So basically, NIBL was very strongly loaded towards the edge of the domain that was forming the stripe. So NIBL has already been shown to be associated with super enhancers. Here we show that the location of cohesion loading may impact the whole domain structure and might lead to a formation of specialized chromatin structure at active regulatory elements. So we confirmed this also by looking at the super enhancer distribution. And this again was following very nicely the location of a stripe formation. So then we wanted to understand whether the CTCF binding at the edge of the domain had any impact on the stripe formation and could help us understand the enhancer activity. So we picked one of the most prominent stripe examples in B cells, which is at the IGH locus. So here we have a super enhancer that is just close to the edge of the domain. So here is the super enhancer, so clusters of active enhancers. Here is NBL profile binding. So you can see that it's enriched towards at the stripe anchor. And here is CTCF binding. And what is really particularly exciting about this locus is the fact that there is a lot of CTCF binding sites. They are all facing the interior of the domain. So we did two different types of experiments, actually three. One in which we have the wild type condition, the complete knockout of the CTCF cluster, and the third one where we left half of the cluster intact. Then the deletion of the cluster of CTCF binding sites results in reduction of stripe formation. And then what was most important for us was to understand whether the reduction of CTCF binding in CTCF is not within the super enhancer, it's just close by to the enhancer, had any impact on the super enhancer function. So to monitor the super enhancer function, we had a look at the transcripts that are generated, that are under control of the super enhancer. So as you can see, we have a reduction of the formation of this, of the production of these transcripts, which is translated in the reduction of the amount of protein that is supposed to be produced at this locus. So the CTCF binding sites at the stripe anchor are important for super enhancer function. So can we relate that in any way to disease? So there is a translocation that brings the IgH locus to MYC and results in formation of B-cell tumors. So we took advantage of a mouse model that we had where we recapitulated this translocation that brings the super enhancer in the close proximity to MYC. And this super enhancer is flanked by our CTCF binding sites and forms a stripe in this B-cell tumors. So when we delete this anchor again, the CTCF binding sites, we observe not only that the super enhancer now starts to activate genes in the vicinity, consistent with the notion that the CTCF binding sites act as insulators, but also it hampers the expression of MYC, which is regulated by the super enhancer, which results also in slowing down of tumor expansion. So this led us to a model where we have two types of regulatory element interaction. One of which is the tethering of regulatory elements by some forces, and we'll discuss it by the end of the talk. And the other element, model is the stripe model, which we propose, in which the location of super enhancer close to CTCF binding sites, so close to the domain boundaries, allows the super enhancer in most probably cohesion dependent way to be tethered actively to regions that it regulates. So we saw before that there were way fewer stripes in embryonic stem cells than in more differentiated cells. So the other question that I wanted to ask in my past was whether chromatin architecture and how it emerges during development. So to model this, I use embryonic stem cells and neuronal stem cells. So basically, we derive embryonic neuronal stem cells from ES cells, and we compared chromatin architecture in these two cell types. So what we found is that chromatin loops are primarily gained during embryonic stem cells to neural stem cells differentiation. And this is visible across the board everywhere, basically. There's a pervasive effect. So it's very similar phenomenon was also observed by others. So we obviously asked whether it's not related to, I'm sorry, I'm somehow, this slides is not properly displayed, but I will explain in simple words what we did. We basically monitored the compaction of domains because the question that we asked was whether this phenomenon is linked somehow to differences in compaction of chromatin during embryonic stem cell differentiation. And it's known that embryonic stem cells have somewhat loose chromatin arrangement. So we ask ourselves a question whether this is not due to these differences in chromatin conformation. And we found that there is no difference. And we used a very nice method developed by Vani Xiang from Jan Ellenberg's laboratory at EMBL, which allows to monitor compaction of chromatin in super resolution. So we identified thousands of loops that are gained upon loss of pluripotency, and these loops were primarily very long range, and loops that are common between embryonic stem cells and neural stem cells are primarily short range. We found that loop formation was related to transcriptional regulation, and loops that were lost were arising upon the developmental step, were primarily arising around loci that are displayed reduced expression, whereas loops that were induced during development are primarily around the genes that are gained in neuronal stem cells. So this led us to a model in which the pluripotent stem cell chromatin conformation is somehow more loose, and loops are not firmly bound to each other, whereas in differentiated cells, loops enclose. They are primarily long range. This could mean many things, and we are actively investigating this phenomenon right now in my lab. So to address this, we use different technologies, for instance, CCAP, which is a very nice technique to monitor interactions between chosen transcriptional regulators and on chromatin using protomics. So in this assay, we basically sonicate chromatin that is cross-linked, and then we focus on our target. So in our case, it's CTCF, and we immunoprecipitate the complexes with CTCF, and then we use protomics to identify proteins that are co-bound with our factor of interest on chromatin. And we did that in collaboration with Professor Jan Krigsfeld from the KFZ and EMBL. And we identified factors that are related to CTCF in embryonic stem cells and neuronal progenitors. And here is just to show you that this technology is really very robust and very reproducible. We identified many factors related to nuclear biology. We also identified cohesions, and we are currently investigating those factors. We want to understand how they impact CTCF function in development, and how can they inform us on the mechanisms that drive the enhanced loop formation during development. So the last couple of slides of my talk, I would like to devote to other classes of loops that we can identify in HiC. So basically, they look like typical loops. So here are those loops that arise as cells differentiate. This is data from a paper by Bonev et al. Very high resolution HiC. I think the highest resolution HiC to date. It's really billions of reads, beautiful maps. And with this very challenging and expensive experiment, the authors were able to, at unprecedented resolution, identify loops and show promoter-enhancer interactions in really high resolution. So there is this kind of loops that do not have CTCF binding sites at both ends, and that connect regions bound by transcriptional regulators, such as, for instance, PAC6, which is important for development of neuronal cells. And these loops, most probably, but we don't know that yet, most probably are related to other than loop extrusion phenomena. So what could be these phenomena? So the recent discovery that promoter-enhancer contacts can be modulated or elicited by the formation of biological condensates could help us understand how this kind of structures may arise. So biological condensates, they arise when two liquids of different properties separate from one another. So this is called the liquid-liquid phase separation. So there is multiple examples of these this phenomenon occurring. We have Cajal bodies in the nucleus, promyelocytic leukemia bodies. Nucleolus is in fact built by liquid-liquid phase separation. To some degree, phenomenon related to this biological condensate formation are responsible for heterochromatin formation. And there is also transcriptional condensates that occur and follow this liquid-liquid phase separation mechanism. So for instance, multiple transcriptional regulators such as BRD4, MED1, transcription factors including OCT4, SOX2, important for embryonic stem cells and neuronal stem cells, they harbor domains that allow them to basically coalesce and form those kind of droplets that you can beautifully see with fluorescence microscopy. So these are domains, the so-called intrinsically disordered domains, so IDRs, that basically mediate the formation of these structures by multivalent interactions. So key features is that they have this capacity to coalesce, to move. They are typically in spherical shapes, and they follow particular fluid dynamics. So right now, there is quite a lot of research put in the direction of biological condensates, how they are regulated, how transcription occurs in the context of biological condensates, and how can they be linked to disease, for instance. So in summary, we heard today that TADs are built by cohesion-mediated loop extrusion. We saw also architectural stripes and CTCF that act to regulate promoter-enhancer interactions. We also saw that CTCF architectural loops increase upon the loss of pluripotency, and these structures frequently demarcate cell type specific genes regulated during differentiation. We also heard that cohesion-based extrusion and phase separation are both implicated in the regulation of promoter-enhancer transactions. So I would like to acknowledge many people that with whom I worked throughout the past years, with whom we basically published the work that you saw, and with whom I work right now. So particular thanks to Wolfgang Huber EMBL, to Rafael Casellas at NIH NIAMS, to Jeroen Kliksveld at DKFZ, also to a collaborator in IDK, Carson Cho. To many people at the Nencki Institute, we started our lab 2 years ago and already established multiple fantastic collaborations. I would also like to thank collaborators from the outside of Nencki, from CENT, from Deutsches Primatzentrum. I didn't really go into details of this project. I would also like to thank RS Aiden, with whom I still collaborate, and primarily now I would like to acknowledge people in my lab that were implicated in various aspects of these projects, and they're constantly right now helping each other and us to understand how chromatin structure is obtained and the basis of astrocyte evolution. Perhaps at some point I will be able to discuss this point in the future. And I would like to thank you for attention, and I'll be happy to take questions.
Thank you very much, Aleksandra. Please unshare your presentation. And now, ladies and gentlemen, it's a possibility to ask the question. And the way of asking the questions is to write the questions in the Q&A form, and then we can ask them to lecturer. Let's have a look. There is the first question. Let me read it. Thanks for a very interesting presentation. So there's a compliment, it's not a question. I understand that we are slowly uncovering the mysteries of differentiation and development. I wonder if there are some studies on comparison of loop formation in various cells.
So yes, there are efforts to study loop formation not only in different cells and different cell types, of course in single cells. For instance, in the lab, we would like to understand the dynamics of loop formation. So we are building systems to monitor loop formation in living cells in live conditions. And there are multiple also attempts to successful to study loop formation across phyla. And Eres Lieberman-Aden is pioneering this work. Also comparing chromatin arrangement in different species like elephants, bees, honeybees, and human apes or whatnot. So there is, of course, efforts to do this, and we are somehow still restricted by prices, by the fact that it's very expensive. So the maps I was showing towards the end from Bonef et al., they had like a couple of billion sequenced read pairs per condition, which is extremely expensive. So that's the limit right now. Hopefully this will be overcome very soon. So.
Okay, thank you very much. Next question. The question is a little bit off topic. Where enhancing sequences are localized, may they be localized in other genes, introns, and if the differences between alleles of the same gene may have different enhancers affecting other genes?
So enhancers are, they are really interesting. They can be located in introns. They can be located, so in principle, an enhancer can act from any place. So there were studies that, for instance, were introducing an enhancer on a different chromosome, and the enhancer was still able to act upon the gene that is supposed to regulate. It's a very complex issue, but in principle, it's possible. There are studies that show enhancers located in exons. And there are studies also that show that promoters can act like enhancers to some degree. So it's quite a complex interplay. So yes, the short answer is enhancers can be found in many different places.
Thank you. And the next question. Again, a compliment. Thank you for a great talk. And the question is the following. Is there any evidence of problems with the loop formation related to age?
Great question. I don't think that we know that explicitly, but it's a very good question, and I think efforts are made in this direction. Yeah.
Thank you. Next question. Is some structural or sequence motif, which is the universal for the most transcriptional enhancers?
No. Short answer is no. And enhancers, they are very cell type specific, much more cell type specific than promoters. So by definition, they have to react to completely different set of transcriptional regulators. Therefore, they cannot be really very similar. So the system evolves in a way that it produces really unique regulatory sequences.
Thank you. Next question. What do we know about plants?
In terms of chromatin conformation, I guess.
So.
Plants have a unique structure. There are topologically associating domains, but it's unclear whether whether, well, cohesins should be acting to form them. There is compartmental interactions. I must say that I'm not really following the field in plants too much, so I wouldn't like to discuss in details this part. But as far as I remember, the chromatin loops made by CTCF are not so evident in plants. But I might be off here. So.
Okay, that's all questions. Aleksandra, again, many thanks for coming to our digital lecture room. This was the first lecture, New Faces of Biochemistry, edition 2.